I'm Mindy Miller. I'm with Terumo Blood and Cell Technologies, and I'm here with Dr. Richard Koya from the University of Chicago. We're here at Advanced Therapies, and Dr. Koya just presented on the 3-in-1 protocol. Can you walk me through what that protocol is?
So we developed a new protocol for activation, transduction, and expansion in a functionally closed system from the start to the finish.
Wonderful. And so as part of this process, this protocol that was just unveiled, what does that mean for the patient? How does that change a patient's journey if they need one of these therapies?
So our development of our protocol started with very few cells, actually 10 million cells, which means we don't even need a patient to undergo a apheresis process. From 10 million cells, we could actually expand towards 10 billion or more cells after 10 days.
That's quite impressive. When we consider the expansion of T cells, it's interesting to know, does the expansion, especially expansion to that level, change the quality of that T cell? Can you talk a little bit about the cell fitness?
Yeah, so that was also an interesting finding. We did a bunch of immunoassays, day 10 of these TCR T-cells. And what we noticed that there is an enrichment of what we call central memory T-cells, okay? And the central memory T-cells actually correlated with better anti-tumor responses and persistency in patients of these T-cells long-term.
Impressive. So with this process, you compared the cells grown on the hollow fiber system to cells grown in a flask. Can you talk a little bit about the two different manufacturing processes and ultimately the results of that?
Yeah, so we have control experiments where we utilize the classical way to manufacture these T cells. And first of all, the expansion was in orders of magnitude better with the hollow fiber system. And as I mentioned, there was a better immunophenotyping profile in these TCR T cells. But also, what is interesting also was that these T cells are more functional, meaning they they destroy the tumor better when you did assays in co-incubating tumor cells with T cells.
Amazing. Thank you for sharing that. When we talk about manufacturing TCR-T therapies, certainly that manufacturing process can be very long, require a lot of labor. How does this new protocol improve that?
So this protocol we develop in a way that minimize the number of staff that we need. Actually, we only one person could do the whole work from the beginning to the end, utilizing this automated. There is a touch panel. We can program the machine, and in comparison to the traditional way, which we require a lot of people specialize in doing the process. This simplifies the whole process and actually minimize also cost with that.
Great. Thank you. I guess one last question. How does this process improve T cell manufacturing as a whole, and what could other investigators learn from your protocol?
So this protocol actually is a way to simplify the whole process and streamline. The bioreactor is a functionally closed system, so the sterility is maintained from the beginning to the end, and the manipulation actually is very straightforward as well. So at the end, we have a high quality product, sterile, which can be compatible for clinical applications.
Wonderful. Thank you for joining me today.
Thank you.